Good morning, everyone, and thank you for coming to the first session. And I have financial interest due to intellectual property and patents in the field, and that conflict is managed in accordance with the University of Pennsylvania policy and oversight. So the background and hypothesis behind much, if not all, of the work I'm going to show you today is that the development of more than 24 antiretroviral therapies and combination therapies to combat HIV has resulted in a dramatic decrease in morbidity and mortality, but there is still significant lifetime costs of a lifetime of antiviral drug therapy. Consider someone infected in their 20s and on these drugs for decades. Further, drug access and compliance issues have contributed to an increase in drug-resistant virus. So the hypothesis then is that we can select and re-engineer either hematopoietic stem cells or T cells, expand those outside of the HIV environment and reinfuse to prevent disease progression and enhance immune function. And so there are a variety of points in the HIV life cycle targeted by gene therapies. There is the initial fusion and entry. There is the reverse transcription, the translation, the formation of the complete vector particle and inhibition of envelope. And I'm going to focus primarily on entry today, although I will mention some other approaches with ribozymes and also finish up with CAR T cells. So due to the time limitations that I have to share work with you, I'll just refer you to this review where we list preclinical and clinical approaches of many other studies that I'm not able to go into sufficient detail of the various points in the HIV life cycle. Okay, so let's talk about editing the genome and in particular why target CCR5. HIV can bind to CD4, but that is not sufficient for entry. That is knocking on the door, but not getting in. To get in, there needs to be expression of a co-receptor, and the vast majority of virus uses the CCR5 chemokine receptor for entry. Now, it turns out that there is a naturally occurring mutation in 1% of Caucasians where they do not express CCR5 on the surface of their cells, and these people happen to be highly resistant to HIV infection, and heterozygotes have a slower disease progression. Now, turning to gene editing, this audience probably doesn't need too much of an introduction to zinc finger proteins and zinc finger nucleases. Zinc finger proteins, there are transcription factors. In mice, there are approximately 500, in humans, about 2,500, but they bind to the sequences of DNA where nature intended, not where we want to mediate therapeutic effects. So Sangamo licensed technology to design zinc finger proteins and conjugate those to bacterial endonucleases, which in a homodimeric fashion can bind to and cleave. And by targeting the specific point in CCR5 proximal to the delta 32 mutation, Sangamo and in collaboration with the University of Pennsylvania showed that the naturally occurring mutation can be mimicked by adding zinc finger nucleases. And actually, this slide should have been before the first, but these binding in homodimeric, heterodimeric fashion induces double-strand breaks. You get deletions and insertions through non-homologous end joining. By supplying a donor DNA, you can engender gene repair, although this is in reality a less efficient process than the insertions and deletions. So how do we take this technology and bring it to patients? Well, that is integrating a process that we developed at the University of Pennsylvania, where we collect white cells by leukapheresis from patients. We can select, enrich, deplete cells, and then we stimulate T cells using an artificial dendritic cell that is a magnetic bead to which two antibodies are conjugated that bind to receptors on T cells, and then T cells start dividing. We can then insert genes at that point using viral vectors or, as I'll mention later on, by electroporating in RNA. We expand the cells, concentrate and wash, remove samples for a quality control test to satisfy FDA criteria specified in the IND. Once those pass and are reviewed, the cells can be released for infusion. And so in yellow are the beads that mimic dendritic cells. They bind to T cells, and when you add the beads to T cells, you can get a logarithmic expansion if you keep adding those beads every couple of weeks. So shown here over 2.5 months of repetitive stimulation is 40 population doublings, and that is pretty close to the Hayflick limit. What we do is we harvest and infuse the cells right here, so there's all the residual capacity to engraft and persist in vivo. So now moving to the preclinical studies, and I'm just showing you a snapshot here. These are human cells that are modified with adenovirus expressing the CCR5 zinc finger nuclease gene, and then injected into non-skid gamma knockout mice, and then challenged with HIV. And what you're looking for is the enrichment of the CCR5 edited cells, because remember, in theory, those have the selective advantage because they cannot be infected by HIV. And in fact, that's what you see here on the right compared to the mock. Now, the FDA asked us to perform extensive studies related to any potential adverse genotoxic events. We performed T-cell receptor repertoire analysis that is shown up top. We performed karyotyping, and we performed extensive animal studies that showed no increase in adverse events or genotoxicity, and that was published in 2013. So here's the design of the trial. We use a chimeric adenovirus to deliver the gene encoding the zinc finger nuclease for each of the patients, enrich for CD4 cells, grow the cells up, and take the samples and perform the testing as I showed on the earlier slide prior to infusion. And this was a first-in-human trial of gene editing, and the goals were to follow safety and feasibility, and also look for engraftment of CD4 cells, HIV viral levels, and the potential selection of the CCR5 edited cells. So here's the design. There were 3 cohorts, a multi-drug resistant cohort, virologic failures. It's actually very difficult to find these patients these days with the antiretroviral drugs available. The other 2 cohorts were immunologic responders to antiretroviral therapy with higher CD4 counts, and failures to antiretroviral therapy with lower CD4 counts. Now, there was an optional structured treatment interruption beginning 1 month post-infusion, and I want to spend a moment explaining why we do a drug treatment interruption. It's commonly used in HIV trials to look at the potential for a therapeutic effect. And what you look for is a delay in virus coming back after the drugs are discontinued, or a reduction in the viral loads compared to what they were before the patients went on drugs. So there's a single infusion at 5 to 10 billion cells, and you can see the endpoints which I mentioned on the previous slide. There's also a separate West Coast study that was run by Sangamo, and I'll show some data not from that trial, but from a follow-up trial later on. So we did see increase in CD4 counts. Remember, these were purified CD4s, and compare that to the levels of CD8s. And we did see in both cohorts where we enrolled patients an increase in the CCR5 gene-edited cells, both in the high CD4 cohort that's shown in red and in the low CD4 count cohort shown in blue. And we also showed that those cells traffic to the rectal mucosa because of sampling and low cell numbers. The quantitation on this is not as high fidelity as in peripheral blood, but I can tell you that it roughly correlated with the levels observed in peripheral blood. Now here's the structured treatment interruption shown in the hatched portion here. And remember, what you're looking for is a delay in virus coming back greater than 2 weeks, or a reduction compared to the level of virus that the patients had before. So the 2 week, you can count and add 14 to 28, which is 42. You can see some patients around the day 56, it's just starting to come back. And the levels at which viral load was present, you can compare the right axis with the colored symbols. Now I want to point out one patient besides the yellow and the orange, and that is the green triangles 205. And this is a patient that remained off drugs for the entire period of the structured treatment interruption, but his virus was undetectable at day 112 prior to going back on drugs. And we're scratching our heads, why did this happen? And it turns out that this was a CCR5 heterozygote. So he had a 50-meter start in the 100-meter dash, and provides some evidence of dose effect and a rationale for the subsequent trials that we in Sangamo were performing. Here's looking at the CD4 count decay, and again, we're looking for the selective survival of the CCR5 modified cells compared to all cells. And all you have to do here is compare the slope of the CD4 counts during the drug treatment interruption. Remember, virus is coming back, so we expect the CD4 counts to come down. And compare this slope to this slope, and you can clearly see that it's different. So in this study, the infusions were safe and well tolerated. We observed durable increases in CD4 counts and CD4-CD8 ratio, showed that the cells engraft, expand, and persisted for more than 1 year in circulation, and that there was trafficking to the gut mucosa, and that there were suggestions of a delay and decrease in viral set point during the drug treatment interruption in a subset of subjects. So what are the next steps after this first-in-human trial? Well, one is to increase the engraftment using chemotherapy, low-dose chemotherapy precondition, such as used to facilitate engraftment in hematopoietic stem cell transplants. Second would be to demonstrate a dose effect by enrolling delta 32 heterozygotes to modifying CXCR4 and to improve the delivery of zinc finger nucleases by electroporation of RNA. So I'll speak first about conditioning with Cytoxan, and this is a study that was performed by Sangamo, and again the rationale is that cyclophosphamide has been used to successfully enhance adoptive T-cell transfer in oncology. So this was an open-label multi-center trial with increasing doses of Cytoxan from 100 up to 2,000 milligrams per meter squared. There was one cohort that did not have CD4 enrichment, a single infusion of modified T cells, and again, the structured treatment interruption. So here's data from that study, and at doses up to 1 gram per meter squared, there were increases in the CD4 engraftment that you can see here. And also that carried over to increases in the CCR5 modified cell engraftment. And what was also observed is a viral load drop from peak after receiving the infusions of the CCR5 modified cells, and that was in 4 subjects that continued on a prolonged treatment interruption. And generally in the field, the consensus is if you see a change of half a log or greater, that is deemed to be significant. So you can see these 4 patients in the red boxes. Now, there was that one cohort that maintained the CD8 cells in the infused product, and that seemed to correlate with decreased proviral HIV DNA. And it makes sense from what we know about how CD4 cells and CD8 cells work together in engraftment and immune response. So the idea is to continue with that scheme with the enriched T cell product, but not separating out the CD8 cells. So here are the conclusions from that study. Cytoxan conditioning at doses up to 1 gram per meter improved the transfer of total CD4 cells and CCR5 modified cells. 3 subjects that were treated with a CCR5 modified T cell product contained both CD4 and CD8 cells that did not affect the safety profile and may have assisted in the goals of the study, which is the effect on the viral load. And further, that large increase in CCR5 modified CD8 cells may mimic what is observed in HIV elite controllers. So next, move to not only preconditioning with Cytoxan, but study enrolling delta 32 heterozygotes and delivering the zinc finger nucleases by RNA electroporation. And this is a study that we've had open at Penn with 3 cohorts. One received zinc finger nuclease modified T cells without Cytoxan preconditioning, and then the other 2 with Cytoxan preconditioning, and then delta 32 heterozygotes with Cytoxan preconditioning. So this study is still open. It is not completed, but I am able to share some interim data with you. Shown up top is the first cohort without the Cytoxan preconditioning. They do receive the structured treatment interruption. We're measuring percent indels here, and we are able to see engraftment and long-term persistence of the CCR5 edited cells. Shown on the bottom are cohorts 2 and 3 grouped together. Notice there is a different scale here, more than double what is shown up top. So that validates what Sangamo had seen with improved engraftment using Cytoxan. And take a note of this patient, the delta 32 heterozygote. You can see much higher engraftment than even we see in cohort 2. And as I said, this study is ongoing and will be presented at future meetings. So next, I'd like to move to some other groups in the field who are taking complementary approaches, different parts of the HIV viral life cycle. And this is a study from Hans-Peter Kiem and collaborators looking at C46, which inhibits the conformational changes required for virus fusion and also has a short hairpin RNA specific for CCR5. And this is being developed by Calimmune. The product is called Cal1, and this is modifying hematopoietic stem cells and delivering autologous HSCs. So the goal is to look and assess for multi-lineage engraftment in blood and the GI tract and see whether there is positive selection for gene-marked cells in blood and tissues following SHIV challenge. And here are the results from that monkey study, and what you can see on the left is in CD3 cells, on the right is in CD4 cells, and yes, in fact, following challenge, you do see an increase. You can see here, this is the control monkey, and these are the two study monkeys, and then here again in the CD4 cells. So another approach that investigators at the University of Pennsylvania are taking is a peptide from the GP41 heptad repeat 2 or HR2 domain, and that is conjugated to the other chemokine receptor that can be used for entry, CXCR4. And what they showed in this paper that was published in November is that HIV entry can be inhibited by soluble peptides from GP41 HR2 domain, and that the peptides could be conjugated to anchoring molecules and overexpressed on the cell surface. So they were conjugated to CCR5 or CXR4, and each exhibited potent and broad inhibition of HIV isolates from diverse clades and tropism. So in other words, whether you conjugate that peptide to CCR5 or to CXCR4, it could inhibit tropic isolates that would bind to either. And so here's the mechanism looking at GP41 and number 1, and then the process of fusion. Here is the conjugation to CXCR4 to block this process and inhibit fusion. So I also want to mention that poster 657 that is being presented tomorrow will show more details on the preclinical studies. We do have a clinical trial that is opening up in HIV-infected subjects testing this approach of a single infusion of CD4 T cells modified with HR2, and the investigational objectives, first, of course, safety and feasibility, engraftment and persistence, trafficking to blood and other sites, expansion and persistence, and looking at the viral load following a treatment interruption. So I want to go back in time a little bit and mention this study that was significant for being the first randomized, double-blind, placebo-controlled phase 2 cell-delivered gene transfer trial. And this was conducted by Ron Mitsuyasu and Johnson & Johnson. 74 subjects received a ribozyme that was a TAT VPR specific ribozyme termed OZ1 or placebo that was administered that modified CD34 autologous HSC. So while there was no statistically significant difference in viral load between OZ1 and the placebo group, there was time-weighted areas under the curve that were significantly lower in viral load and higher in CD4 counts. And if you look at this figure, it's so tantalizing but a little frustrating because if that group had gotten, say, a little bit higher dose, it would have reached that half-log profile. But again, comes back to the point that I mentioned up front, which is looking at efficiencies in gene modification and the appropriate dose to see a therapeutic effect. And finally, I want to mention this study before moving on to the first trials in HIV. And that was a study performed by Dave DeGusto and John Zaya at City of Hope. This was modifying CD34 cells in patients undergoing transplantation for AIDS-related lymphoma. And I call this approach the triple threat because it had 3 RNA-based anti-HIV approaches: a TAT-REV short hairpin RNA, a TAR decoy, and a CCR5 ribozyme. There was successful expression that declined, but because of the ethical requirements of design in this study, not only were the gene-modified stem cells administered, but non-gene modified cells were administered that probably diluted the ability to track the cells. Nevertheless, engraftment could be observed as shown here, one also hopes that in future trials we can proceed with higher doses of cells and new constructs that allow for increased potency. So how long can gene-modified cells persist in vivo? Well, I take you back to the very first chimeric antigen receptor T-cell trials that were conducted by Cell Genesis. And this was a CAR that was a little bit different. It was not an antibody on the outside, but native CD4 that bound to GP120, that is the mechanism for the specificity. It was a first-generation CAR. New generations have co-stimulatory domains integrated into the CAR design. And because of FDA requirements for 15-year monitoring in gene therapy studies, samples were collected by Cell Genesis that we analyzed for long-term persistence. And much to our surprise, we thought we would see a drop-off after some months or a couple of years. We saw a relatively stable persistence going out 7, 8, 9, 10, even one time point at 11 years. And when you do the statistics on the first 9 years of time points, this turns out to be a 14-year half-life following a single infusion, which is really quite remarkable. We did RT-PCR and were able to detect transcripts in all of these patients. These were at the limit of detection, so we know that gene is not silenced. And going back even further to 1990, the first 2 patients treated with any kind of gene therapy, the SCID-ADA kids, we know that those persist for, shown in this report for some years ago, this was 10 years after, but those cells have persisted even longer. So this is a selective situation. We don't know how well this correlates to cancer and to HIV. So at Penn, we have treated close to 500 patients with gene-modified T cells in HIV or in cancer, and we now have over 1,700 patient safety years with the absence of genotoxicity modification in T cells. And I close with, why is this man smiling? Because this is the only man that has been thoroughly and completely cured of HIV. This is Timothy Ray Brown. He is, or was, an HIV-positive patient that was diagnosed with AML, relapsed after chemotherapy, received a stem cell transplant with CCR5 delta 32 negative cells, and he is now HIV-free. So if we can mimic this situation for patients who don't have to undergo stem cell transplants, that would be a wonderful thing for patients. And where do we go from here? It is, I think, the emergence of combination antiretroviral drug therapy points to the requirement for multiple points of attack and defense. That's why the triple threat approach of City of Hope and others that are complementary, I think will bode well for long-term potency and also persistence. And I think the studies that I've shared with you today, as well as others, point to the importance of delivering a sufficient number of gene-modified cells. So I'll close now and thank the collaborators at Penn, at Sangamo, the NIH and NIAID for our funding, and also to collaborators and colleagues whose work I was able to share with you briefly, and again apologize for any omissions due to time. So with that, I'll stop. Thank you for your attention, and happy to take any questions.
Thank you for that wonderful talk. I think we have time for just one question, if there's something very quick. Otherwise, I think we'll go ahead with the next speaker.
Can I just ask, what was when you infused your gene-edited T cells, what's the phenotype of the cells infused, and also when you've analyzed patients long-term who have got T cell persistence, what cells actually persist?
Yeah, so actually the phenotype in vitro after expansion is somewhat artifactual because in some ways, for example, the expression of CD45RO, they all are RO positive, 100%, but they also are significantly portion CD62L positive. And we believe that assists with engraftment. And we know with limited data from HIV, but much more from our studies in cancer, that there is a burst of effector T cells. And after that peak and the long-term plateau, those cells either revert to central memory or central memory cells persist. So with the CD4 zeta trials that were conducted by Cell Genesis, you can think of that repetitive restimulation, right? Because you have CD4 cells, they're coming out from the bone marrow, and that may be a factor in the long-term persistence, and also something I think to consider for other approaches. How do we get these cells to persist long-term if virus is controlled with drugs and there isn't significant restimulation through viral antigens?
Thank you. Okay, thank you very much. And so
I thank the organizer for inviting us to present our work. So as you know, TIGET in Milan developed different gene therapy approaches to treat primary immunodeficiencies based on autologous infusion of CD34 positive cells transduced with a retroviral or antiviral vector. And over now the past 10 years, actually more, our group was committed to study the integration site distribution of this vector in our gene therapy treated patients. And the first goal was to address the safety of the gene therapy clinical trials, but more recently we exploited these tools to address more basic biology questions regarding the dynamics of the clones that were infused in vivo in our patients and the hierarchical relationships among the cell lineages. And the main workflow and the concept behind these studies is the fact that each infused cells that is transduced carries a unique cellular marker in that we carry a unique integration site. And by sampling the infused cells from the patients at different time points after gene therapy and isolating different cell subpopulation, we can retrieve these unique cellular markers, and we can, with LAM-PCR or whatever other methods for vector genome junction enrichment, and combine with Illumina or whatever other NGS sequencing method, we can identify these clones through the identification of integration sites. So we can have an identification of the clones that is retrieved to the identification of the locus where the vector integrates and the single closure genes, but we also have a surrogate marker of clonal abundance, in this case represented here the sequence count related to each integration site. So we have a clonal identification and a clonal abundance measurement. And we can use these two data for performing tracking and cell hierarchical analysis in the patient. So over the past years, we published different works related to this kind of study. And I'm going to focus here on mainly the two work that were directly focused on this study. Although the first work that is reported here, even if it was focused on safety and efficacy of gene therapy, already contained some information regarding the tracking of the infused cell clones in viscoodexinone data. And I will also show you some data that are not published yet and that will be contained in a manuscript that is under preparation now. So the first application of integration site to track individual clones in patients that we performed was in the context of a confined subset of hematopoietic cells, in particular T cells. So what we decided to do is to isolate the four main T cell subsets, naive, central memory, effector memory, and effector memory positive cells, from two sets of gene therapy treated patients that were treated for ADA SCID. In the first set of individuals, which was a first early gene therapy trial, patients only received peripheral blood mature T cells. In the second trial, which is now the one that achieved the highest efficacy, patients actually were treated with bone marrow CD34 positive cells. And in particular, in the first trial, these patients, they are missing hematopoietic progenitors and precursors. So what happens is that if you detect a T cell in these patients many years after the infusion, these T cells, they must have survived for a long period of time in the peripheral blood. So particularly in the first clinical setting, we could have, thought that we could have very good information regarding the T cell survival in absence of any production from an upstream progenitor. So what we did very simply at the beginning, we isolated this four subpopulation and we collected the integration sites. And here came out surprising results. So on the left, you have the data regarding the hematopoietic stem cell gene therapy, where you can see, as you can see from the gel and from the Venn diagram below, we collected, although with a low throughput early analysis, integration site from all the subset and also from naive T cells, which makes sense because these patients have progenitors on top that are keeping producing the lymphoid precursors. What was surprising was that we detected the vector-positive naive T cells also in the second set of patients, which were the patients treated only with lymphocyte gene therapy. And we collected vector-positive naive T cells up to 10 years after last infusion. So the question was, how could naive T cells survive that long in patients preserving the naive phenotype? Because the main concept is that naive T cells are short-term surviving, actually. So we should have found only memory cells in these patients with a vector. And if these naive T cells were present in vitro before infusion. So we kept this question on hold for quite a while. We thought that there was some issue of purification of cells, or purity of the analysis, until this paper came out in 2011, which described a population that carry a naive-like phenotype, but have also some memory marker, in particular CD95. This population was described as T memory stem cells, and it was described of having a T cell precursor activity, but being still capable of long-term surviving. So we came back to our patient sample, and then we asked, maybe the cells that we previously defined as naive T cells, in particular in our PBL gene therapy patients, maybe these cells are actually T memory stem cells. So we went back to the patient sample, and this time, instead of sorting only naive T cells, we also sorted the T memory stem cells, which are here reported in red as TSCM. And these cells were sorted from the double positive compartment, CD62L positive, CD45 RA positive. And what we actually found is that in the PBL gene therapy, as you can see on the panel on the right, the vast majority, if not all, of the cells that were contained in the precursor compartment were indeed T memory stem cells. So what we previously thought to be naive T cells were T memory stem cells. So at this time, we had a tool to identify integration site in this newly defined subset and to understand how long T memory stem cells can survive in a human being without the supply of hematopoietic progenitors. And that's what we did by collecting integration site from PBL gene therapy treated patients on the left and from, as control, hematopoietic stem cell gene therapy patient treated for this kid on the right. And we isolated the integration site both before infusion and after infusion, in particular in PBL gene therapy patients. So we use integration site sharing as a surrogate marker of the precursor activity. What does it mean? It means that the higher it is the number of integration that a given cell type shares with the other, the higher should be their position in the hierarchy. So they should be on top of the hierarchy and generating the other cell subset. And that's actually what we found in PBL gene therapy patient, both before infusion, where we were able to assess that there were no naive T cells at the time of infusion, but still there were some T memory stem cells there that were infused. And actually, even in vivo, many years after the last infusion, we still detected T memory stem cells that were sharing integration with the other subset. You can see from the ribbon graph on top, each ribbon is proportional to the level of sharing of integration site between the groups of cells that are reported as colored there, and then the quantification of the sharing is reported on the bottom. So the T memory stem cells were the cells that had the highest integration site sharing, so were on top of the hierarchy, which makes sense with the precursor capacity. When we have instead naive T cells around, because we have lymphoid progenitors in the bone marrow, the naive T cells are on top of the hierarchy, as you can see from the bottom right panel. They share the highest number of integration, followed by T memory stem cell, central memory, and effector memory. And these data were very consistent with the linear model of the T cell differentiation that was proposed by Cd4 and other colleagues. So our integration site have a biological meaning. So we can rely on this to perform hierarchical analysis. And the second question was, can we track identical clones within the T memory stem cell subset over time? So are there clones that retain the T memory stem cell phenotype and then can be tracked at two different time points? And we actually were able to detect a few clones that were detected both at 7 and 12 years after the last infusion, that retain the T memory stem cell phenotype, which I show here in this heat map, but they could also differentiate in the other subset. So we had a measurement of the survival and of long-term differentiation capability of these cells that are endowed actually with some sort of self-renewal capacity, and this is why they are defined as stem cells. So with this work that we published on Science Translational Medicine, we were able to provide a proof of principle that integration site analysis is a valuable tool to provide answers to questions that are related to the basic biology of the cells that are engineered and infused into the patient. So we then extended our study to the whole hematopoietic system. And the context in which we performed this study was a gene therapy trial for Wiskott-Aldrich syndrome that was introduced very well from the previous speaker. And I want to mention only here that this trial was a particularly, let's say, lucky clinical setting where to perform this kind of study, because this patient had displayed a very persistent multilineage engraftment, and they did not display any major clonal skewing in any of the lineages that have been analyzed. So it's let's say, I would say the optimal setting where one could perform a study of hematopoietic reconstitution directly in human being. So the first goal was to make sure that we were selecting and purifying the cells with a high degree of purity. And what we did is that we isolated from this patient at different time point after gene therapy, different cell types through different selection system, either magnetic beads, FACS sorting, and actually we confirmed our data through the integration site that were derived from colonies that were grown ex vivo. And what we did is that we basically scanned at different time points after gene therapy the reconstitution of the hematopoietic system in these patients, collecting data not only from the peripheral blood, but also from the bone marrow. So we had access in these patients to bone marrow aspirates at different time points after gene therapy, and at the same time from peripheral blood samples. So we could cross-compare what was going on in the bone marrow and what was going on in the peripheral blood. And we tracked more than 89,000 clones from 7 peripheral blood and 7 bone marrow lineages over time, including the compartment which is composed by bone marrow CD34 positive cells. So the first things that we want to look at was at the overall clonality of the hematopoietic reconstitution through a diversity index. On the left, you can see the diversity of the peripheral blood cells in the 4 patients that were analyzed over time. So you see after an initial phase, there is a stabilization of the diversity. So again, to testify that we have pretty polyclonal and stable repertoire in this patient. And we could then further dissect this information, analyzing the diversity of each individual lineage over time. And in the panel on the right, you can see in red the diversity of bone marrow CD34 cells over time. You can see that we have we could immediately see that we have 2 different phases of hematopoietic reconstitution. So we have a first wave that lasts more or less until the first 6 months, and then after 12 months, we have actually a pretty stable, we reach a pretty stable diversity that is then maintained over time up to many years after gene therapy. So we then wondered what was going on in the early versus this late phase. And the first question was, can we track clones in the early phase that are persistently surviving over time, or the clones that are active in the early phase tend to expire? So what we did is we analyzed individual clones isolated from the first 3 months after gene therapy that are reported here in this heat map in the bone marrow and the peripheral blood. These are the top 5 clones in terms of our presentation as an example. So each row is a clone, and the color is the, let's say, the proportional to the size of the clone. So as you can see, the clones that were isolated at 1 and 3 months were poorly, if not at all, detected later on. And we have like some evidence of clonal succession in the first 3 months after gene therapy. What happened is that after 6 months instead, we start to more consistently recapture identical clones over time. This is pretty stable over a long period of time. So we had the first clue that there are 2 different sets of clones that are active during reconstitution. And the recapture capability of the clones after 12 months allowed us to use a system that are usually used in ecology to assess the overall abundance of the population by capture and recapture methods. And this provided us the first tool to let's say, estimate how many clones were composing the engineered hematopoietic system. And the general take-home message here is that after 1 year, only a few thousand clones seems responsible of the production of the whole hematopoietic system out of the millions of cells that are infused into the patients. And this actually was consistent with the data that were collected both in murine models and in non-human primates. The second question was, since we have the data from the CD34 cells in the bone marrow and at the same time data from mature cells in the peripheral blood, can we measure the output of the CD34 cells over time? So we envisage this visual representation in which I would like you to focus on the rainbow part as compared to the red part. The rainbow part represents the number of shared integration from CD34 cells. So the bigger it is this part, the higher it is the output of CD34 cells, and the more colored it is, the more diverse it is. So it means that it goes through all the different lineages. And let's see how this output changes over time. So in the first month, we can have different starting situation of a constitution in the patients. But what we observed is that at 3 months, we detect in every patient a shrink of this clonal output from CD34 cells. So there is like an exhaustion of the first wave. Then after 6 months, there is a takeover of what we believe are the real long-term hematopoietic stem cells that, as you can see, preserve a very strong, very consistent multilineage output over a long period of time. So again, we had also a functional, let's say, proof of the existence of 2 different waves of hematopoietic reconstitution. So we came out with this model of hematopoietic reconstitution after transplantation in humans, which is basically consisted by a group of short-term HSPC, which are very active in the early phases with a limited multilineage output, which decreases up to 3-6 months. And then at 6 months, we have the takeover of the long-term hematopoietic stem cells. So we decided on the basis of this working model to work a little bit more in detail, and now I'm going to move to data that are not published, and to further dissect the CD34 composition and repertoire in these patients, and in particular to isolate hematopoietic stem cell progenitors and mature lineages, and focus on the comparison between these different stages of hematopoietic reconstitution. So what we did is that, again, we combine, in a work, we combine a phenotypic characterization of the CD34 compartment in this patient, isolating 7 major hematopoietic progenitors, HSC, MPP, and MLP from the CD38 negative compartment, pre-BNK, CMP, GMP, and MEP from the CD38 positive compartment, from these individuals at different time points after gene therapy, and we combine this with the molecular tracking methods based on integration site analysis. So having access to the phenotypic characterization of a bone marrow of a patient after transplantation is not obvious. Actually, I cannot recall many groups that have access actually to bone marrow aspirates after transplantation. So we don't really knew until now what's going on in the bone marrow of the patients after transplantation. So the first novel information from this work regarding the phenotype of the bone marrow. So we first collected as a reference the bone marrow CD34 composition from adult healthy donor and pediatric healthy donor, as you can see here from this bar graph. And in particular, the white fraction of this bar graph contains CD34 positive lineage positive cells, which I can tell you are mostly CD19 positive, so B cell precursors. And as you can see, you already can appreciate differences between the composition of an adult bone marrow and the pediatric bone marrow, which makes sense with what is known in the literature, the fact that in the pediatric patients there is an enrichment of B cells precursor. Then let's see our patients, and let's see what was the situation before gene therapy, as you can see in the third column, which this patient have an even higher contribution of lineage positive, CD34 positive cells, and actually a slight enrichment in the pre-BNK fraction, which is the green one. Then we sample the bone marrow at different time points after gene therapy. So in the first month, as you can see, we have an enrichment of composition of pre-BNK and lymphoid T precursors as compared to what is control and what was the situation before infusion. Then in 90 days, these first waves start to renormalize, and then, as you can see, up to 5 years after infusion, now the distribution of the composition of the bone marrow is pretty stable, and is somehow recalling what it is expected from a pediatric matched healthy donor. So again, this information is suggestive of these two waves of hematopoietic reconstitution. So we added to the tracking and to the functional validation of the wave, also the phenotypic validation of this wave in the bone marrow. So if we can focus, for example, specifically on the number of cells per microliter in two major progenitor compartments, the CD38 negative, these are the very primitive cells. As you can see, there is a first peak of contribution by MLP and MPP, while HSC, as you can see, they remain pretty much stable. Then after this first peak, this contribution decreases to levels that are not exactly matching what is in a normal individual, as you can see from the latest dots that are on the very right of the graph, that are put there as a healthy donor reference. But this level is pretty stable over time. The same can be said for the CD38 positive compartment, where we instead observed a boost of GMP and of ATP, in particular when neutropenia needs to be resolved after conditioning, and when there is the need for the production of lymphoid cells, so in the very early phases. So this is very consistent with what is expected in terms of reconstitution, and as you can see, they reach levels that are pretty similar in this case to the healthy donor contribution. Second question is, can we then isolate these cells and first of all detect whether they are gene corrected? What is the vector copy number of this in these cell types? And we isolated the different differentiation paths. So when we look at the T cell compartment, we see the HSC, MPP, and MLP, they have a relatively low and stable, with the exception of only one outlier, contribution of vector-positive cells. And we can see the effect of what is the selective advantage for gene-corrected cells only in the CD3 peripheral blood cells, so very late in the T cell development. For the B cell compartment, we have different stages of differentiation. So you can see again, up to MLP, the level of vector-positive cells is pretty stable and low, but for an MLP to become a pre-BNK, it needs the therapeutic gene in this patient. So we start to have a first selective advantage, and which is further evident for the differentiation into CD19 positive bone marrow cells and CD19 positive peripheral blood cells. So we can really track the effect of selective advantage in these patients. And in the myeloid compartment is some sort of control, because we don't expect this patient don't have any major myeloid defect, so we don't expect a selective advantage for therapeutic genes. And actually you can see that the vector copy number is pretty stable there, although we observed a slight decrease of vector positive cells from the CMP to the GMP state, which then is stable when the cells differentiate into CD14 and CD15. So again, we put a huge effort on analyzing integration site in all these different subpopulations and cross-compare them with the integration site in the bone marrow and the peripheral blood lineages. We now collect, actually this number is now higher, but let's say more than 15,000 integration sites from the different progenitors at different time points after gene therapy. And I would like to give you three key messages that we are deriving from our data, and but we have much more analysis going on, and I don't have time here to show you everything that we are doing for these studies. The first message is now we are collecting integration site at early and late stage after gene therapy. So we wanted to do a perspective HSPC clonal output tracking over time. So we first look at the HSPC that were isolated at 2 months after gene therapy. In this heat map, they are represented in the row, the 7 different subpopulations, and at the bottom of the heat map, there are present all the other subpopulations. So where you see color signal, it means that we detected the sharing, and the intensity of the coloring means that there is proportional to the intensity of the sharing of each subpopulation with what is on the bottom. So you can see that these HSPC that were isolated 2 months after gene therapy were pretty active at 2 months. They kind of get shut down at 3 months, and then they become active again at 6 months, but with a mild activity up to 3 years. If we compare this with HSPC that are instead isolated at 3 years, and we trace back where we were able to detect them, you can see that they were basically not active at 2 months. And they start to be active only at 6 months, and their activity is much stronger as compared to the HSPC that were isolated at 2 months. So we have clearly here 2 different sets of HSPC. So the first one that became activated, they were able to be shut down for a period and then to reactivate, but to a milder level, while instead the one that activated after 6 months, they were the one that were sustaining with a very strong output the hematopoietic production over time. But these cells were basically silent or very poorly active in the first phases. The second message is that we can use integration site similarity also to track the hierarchy within the CD34 compartment. I don't have time to show other more, let's say, detailed analysis, but I wanted to leave you with the message that actually the sharing of integration site between primitive and more committed progenitor makes a lot of sense with what is known in terms of biological differentiation. For example, as you can see here, each arrow is proportional to the sharing of integration site, and the thickness of the arrow actually is proportional to how many integration sites are shared between the lineages. And you can see, for example, in these 2 patients that MLP are sharing a lot of integration site with PreB and K, which makes sense with the differentiation path of these precursors. And I can tell you, not from this data, but from other data that we are collecting, that we were able to estimate that on top of the hierarchy, at steady state, the HSC are the ones that are responsible for the production of the whole hematopoietic system, which is something that is somehow contrasting with what was proposed in some published work, where they claimed that the MPP actually is the one that is more active in the steady state hematopoietic production. So I wanted to leave you here and tell you that we are doing other studies also on the heterogeneity on the hematopoietic reconstitution, but I don't have time to show you the data here. I hope I convinced you anyhow that integration site analysis is a tool to provide a very relevant biological question in directly in human beings, and we actually could, with our study, assess the persistence and the activity of engineered T-cell clones, the survival potential of T memory stem cells. We could track the population dynamics after reconstitution and at steady state, and the contribution of individual HSPC progenitors. And this work was performed at TIGET, and I would like to thank my former group there at TIGET, particularly Serena, Luca Vasso, Ricci, and Francesca Dionisio, and Alessandro Iuti, which is the co-PI of this study. And we are now moving these and other studies, similar studies, to our new lab, which has been set up in the past year in Boston, where I'm working with Cristina Barricordi, Mariano Perfito, and Danilo Perin on a series of these kind of chronotracking studies in different diseases that are treated there, and in collaboration also with other groups as well. So I would like to thank you for your attention. I'm happy to take any question. Thank you.
Thank you, Luca. Wonderful talk. Thank you.
Thank you.
Cindy.
You can shout.
Whatever, I can talk loud, and actually Bruce can maybe answer part of my question, not me. So the T memory stem cell work, really interesting, but ADAs can obviously not a normal environment. It's a metabolic disease. I know they're clinically metabolically much more normal, but has anyone looked, to your knowledge, or are you looking at these different T memory stem and other compartments in some of the CAR trials situations. Also, obviously not normal when you're putting in a chimeric antigen receptor necessarily, but I think that'd be really interesting. And then the second question or comment is, when you look at the bone marrow early after transplant in our primate studies, as you presented at ASH, and hopefully eventually we'll publish, you have incredible geographic segregation of HSPC clones in the marrow, and it takes a really long time for that to mix. So when you're looking at 2 months, I just don't believe that you necessarily know what's going on in the whole HSPC compartment. And I know in children on gene therapy trials, you're not going to be doing multiple bone marrow aspirates, but in our monkeys, we found that you cannot know what you're looking at in the whole bone marrow compartment if you just look in one site. And until you get mixing, which is really can be very late, up to a year, it's very hard to interpret that. So it's one caveat about the marrow studies.
Okay, regarding the first question, yes, we are doing this in other gene therapy settings. Actually, we are collaborating with the London group on the Skidix1 gene therapy, and we are also establishing collaboration. We will do the very same T-cell tracking studies also in CAR T-cell related trial. The second question is true. Actually, we cannot really say whether the topological distribution of these cells is influencing this data. What we can do is to analyze the myeloid production in the peripheral blood. That's what we did. We actually didn't have time to show you here. So the myeloid production should somehow normalize for this different distribution, because it should tell you, since these cells are widespread all over the peripheral blood, it's much easier to have like more evenly distributed contribution of integration site from different sites of the bone marrow, in particular in these initial phases. So if we use myeloid cells data as a surrogate marker of the bone marrow precursors in the early phases, we can get around on this. But obviously we are very curious to see in a setting in which you can have access naturally to more sites if the data are matching.
So congratulations on wonderful data sets and presentation.
Thank you.
To answer the stem cell memory question, yes, we're looking at that. Joost Mellenhorst from Penn is giving a talk today. He can answer what he's going to show, or you can just wait and go to his talk, which may be even better.
Sure.
But the question I wanted to ask is the transition to steady-state hematopoietic stem cell reconstitution. And given the sampling limitations of bone marrow and peripheral blood, and I can't remember how many patients in which you've done the analysis, but is there any correlation or trend you've been able to identify as far as cell dose delivered or age of the patient or other factor that can give you a sense for accelerating that or having the most robust reconstitution?
I guess here we are in the context which was pretty uniform in terms of number of cells, ages, and reconstitution. So we cannot really stratify our data for this very interesting parameter, I must say. So I would say that we didn't observe a major difference. Now we extended the study now up to 7 different treated patients, but these were all Wiskott-Oldie syndrome patients only in our setting, and basically the age and the dose of the cells was very similar, and they end up with a very similar kinetic of reconstitution. 2 of them received mobilized peripheral blood, CD34 cells, so we are digging into this, so the cell source could be an influence. And one of the things I'm going to do in Boston is to cross-compare along the different gene therapy trials and the different conditioning regimen, ages, and cell dose, the different reconstitution kinetics in these patients. So we're going to be able to address this, but so far in our court it's too uniform to stratify for this information.
Very interesting talk indeed. I enjoyed it. We are doing
Thank you.
integration site analysis
Appreciate
in
it.
CAR T cells. We discovered a unique integration and an epigenetic regulator that actually affects the kinetics of the cells and potentially contributes to the therapeutic effect of CAR T cells. So one of my questions really is, have you seen any of the integration sites in or near oncogenes? Do you have any suggestions that the integration sites contribute to the differences in pharmacokinetics of the cells? And then the other question I have is even more technical. I know of your 2015 Science Translation of Medicine paper, which absolutely beautiful work.
Thank you.
I know there are challenges in cell numbers and getting enough integration sites analyzed. So how do you make sure that when you analyze stem cell memory T cells that you have enough numbers that you actually have a full repertoire to analyze and compare with the rest of the T cell compartment in 4s and 8s and subsets?
Yeah. So regarding the first question was about the, so first of all, I would like you to ask you a question.
Yeah.
Which kind of vector are you using, a retroviral or lentiviral?
Yes.
Okay. So in our, this is a retroviral vector. We published in 2011 the differences between T cell and hematopoietic stem cell distribution of the retroviral vector. So when you transduce a T cell and a hematopoietic stem cell with the retroviral vector, the vector goes into different places. What we observed is that we do not target at all LMO2 in T cells. We do not target MECOM area. We observe some genes that are related to the stemness, but it's difficult to dissect whether these are proto-oncogene or stemness gene. So far, we didn't observe major skewing that might suggest the effect of integration site on the selection of T cells, although the follow-up is, the number of patients available and the follow-up is limited. I could tell you that it could definitely be a case. There could be definitely an influence on that. Also in our setting, I would say more on the influence of the genomic environment on the therapeutic gene expression. So depending on where the vector lands, you can have a higher therapeutic gene expression, so these cells are naturally selected because they are more active and they can actually differentiate better. Regarding the second question, can you remind me the second part of it?
More of a technical question for cell numbers. So you're analyzing the various subsets. Do you do a in vitro enrichment, expansion?
We try to avoid that because this would induce some other selection and kernel skew that we want to avoid. Right. We struggled for this, and we end up with quite a good method for amplification of genomic DNA at the time of sorting. So we sorted the cells, so we could go down to even 600 cells for some patients, but with whole genome amplification, we could have enough DNA material to retrieve both vector copy number data and integration site data. And it's something that we also translated for the study on hematopoietic stem cell progenitors, which in some cases there are even fewer cells.
Okay, thank you very much, Luca, and thanks to all the speakers for their talks today, and to you for your attention. Thank you very much.